friends to a new session on genomics and proteomics. In this session, we will discuss genetic and physical mapping, long range and global physical mapping, integration of genetic and physical maps. Genetic and physical mapping. Gene mapping describes the methods used to identify the locus of a gene and the distances between genes. Genes can be viewed as one special type of genetic markers in the construction of genome maps and map the same way as any other markers. Molecular mapping can be of following two types: genetic maps and physical maps. Both genetic and physical maps provide the likely order of items along a chromosome. Genetic map serves to guide a scientist toward a gene, just like an interstate map guides a driver from city to city. Physical maps mark an estimate of the true distance in measurements called base pairs. Therefore, allow scientists to more easily home in on the location of a gene. Genetic and physical maps illustrate the arrangement of genes and DNA markers on a chromosome. The relative distances between positions on a genetic map are calculated using recombination frequencies, whereas a physical map is based on the actual number of nucleotide pairs between loci. These maps are key source for understanding genome organization. It is a critical step in identifying the genes involved in a genetic disease. Once a disease gene is accurately located, we can determine its DNA sequence and study its protein product. For example, cystic fibrosis is the most common lethal inherited disease in the United States. As many as 1 in 2,500 Americans of Northern European descent carry a gene with CF. In 1985, the gene was mapped to chromosome 7q31 to q32 by linkage analysis. 4 years later, its DNA sequence was determined by Francis Collins and colleagues. Now, we know that the disease is caused by the defect of a chloride channel encoded by the CFTR gene. Genetic mapping. Genetic mapping is the process of determining the order of and relative distance between genetic markers, specific sequences or heritable elements that generate phenotype on a chromosome based on their pattern of inheritance. Genetic mapping in bacteria relies on chromosome transfer between cells. Genetic maps have been used successfully to find the gene responsible for relatively rare single gene inherited disorders such as cystic fibrosis and Duchenne muscular dystrophy. Genetic maps are also useful in guiding scientists to the many genes that are believed to play a role in the development of more common disorders such as asthma, heart disease, diabetes, cancer, and psychiatric conditions. Mapping also provides clues about which chromosome contains the gene and precisely where the gene lies on that chromosome. The basis of genetic mapping: linkage analysis. Genetic linkage analysis is a powerful tool to detect the chromosomal location of a diseased genes. It is based on the observation that genes that reside physically close on a chromosome remain linked during meiosis.